everyone, I'm Ruba Bayram, a first-year medical student and a member of PACademia. Today I'll be discussing the chapter Transplantation, which is going to be the second part of the first video, hypersensitivity and transplantation. Okay, so let's dive deep into it. First, before we start, we need to understand a few terms. So transplantation, in transplantation, I have a person, right? I have another person, and I want, let's say, a kidney. So I don't have a kidney, no kidney. This, yes, kidney, right? And I want a kidney, so I will get the kidney from this person. So here, this person is very kind to donate their kidney to me. Okay, so this is the kidney right here. Okay, okay. The person who is going to be getting the kidney is what? Is going to be called the recipient. It's going to be receiving. The person who is going to be giving it is the donor. He's the one donating. And then this kidney, the small kidney, is known as the graft, the thing that is being donated. Okay, so this is the picture. Who is receiving the kidney? The recipient or the host. Who is giving me the kidney? That's the donor. The kidney, what is it called? The graft, because it could be any organ. It doesn't have to be kidney. Okay, so that's that. Now there's many different types of grafts. Here, the four main ones. Autograft means what? From one person, from me, and I'm giving it to me. Yeah, I need, let's say, a transplant. I was a burn victim, let's say. So this patient is a burn victim, and they have lost some of the skin that is on their arm or something. So they will take skin from their leg, for example, and put it here. The graft is my own. The donor is me. The recipient is me. Everything is me. So that's why it's autograft. From one part of the body to another, right? Like from the trunk to the arm. That's an example, like in burn patients. Okay. I broke a tendon or something. I'll take a tendon from another place. A muscle. I'll take a muscle from me, put it to me, and then the thing is from myself. So everything, the recipient, the graft, the donor is me. That's why it's called as autograft. Auto means self, right? And the isograft is going to be between genetically identical individuals. It's basically the monozygotic twins. This is the main example. So isograft is between monozygotic twins. Two twins, they're going to be what? Donating between each other. They're twins. And they're monozygotic because they are genetically identical. If they were dizygotic, it won't work. Because as we know, dizygotic is what? Dizygotic, they are genetically different. So this example is monozygotic twins. Okay. Allograft. The allograft is between different members of the same species. I have a species. Within a species, I have many different members, right? An example is between two different people. Me and someone else, like in this case, right? These are two different people. This is person number one, this is person number two. But they are of the same species. Why? Because both of them are humans. Both of them are the species, which is the human species. Do you understand? So this is allograft. The other type, like xenograft, no, they are going to be between members of different species. So basically, a pig with a human. Like I'm a human and I need let's say something, like a kidney or whatever. I'm getting it from a pig. Is the pig the same species as me? No. They're not the human species. So this is the xenograft, between two different members or two members of different species. So autograft, everything is from me within myself. Isograft is genetically identical. Main example you think of, monozygotic twins. The allograft is going to be between members of the same species. Yani example, the main one, human to human. The xenograft is going to be between what? Members of different species. So a human with a pig. Now, this is the types that we mentioned. The allograft and the xenograft, these are the ones that you're going to be mainly going to be rejected. Because why? Because they're from two different things. So the allograft and the xenograft, for example, here, they're between two different people. So there's going to be some differences. And xenograft even worse. The differences are probably going to be a lot, right? So here they're saying the rejection is probably going to be happening mostly between these two. Okay? But the autograft, why will you reject your own thing? The donation, the graft is yourself. The donor is yourself. So why would I have any rejection towards it or any reaction towards it? The isograft, they're genetically identical. Why would I? So the allograft and the xenograft are the ones that are going to be having the genetically identical. Okay, and you can see here the arrows. It's a great way to show you the arrows. For instance, here they're showing you this, for example. What does this arrow indicate? Is that within myself, the isograft, these are two similar people. You can see they're the same color, so they're genetically identical. That's how they're trying to show you. The allograft we mentioned is between two different humans, right? Because it's the same species, but two different members. So you can see it's between this and this. And you can see they're different colors. So they're showing you that, okay, they're not genetically identical, but they're still a human, but they're just not genetically identical. Here we can see that's an animal and it's giving it to the human. So the arrows are a great way to actually illustrate it as well. Okay. Now, we mentioned in the allograft and xenograft, we can have some sort of rejection. There is something known as the host versus graft response, and then graft versus host. Let's talk about host versus graft. The first one is the one that's attacking. So I have, for example, host versus graft, and I also have graft versus host. The first thing is the one that's attacking. In this case, host versus graft, who's attacking who? The host, which is me, is attacking the graft. The graft is what? The donation, the donated organ. So I'm attacking the organ. This is in the case of rejection, for example, here or here, right? And the graft versus host, who came first? The graft. So the graft is the one that's attacking me. Okay? So here that's what we're saying. Host versus graft is what? My body will be the one that's attacking the organ that I received. Why? Because host is the first. So the host is attacking what? The second thing, which is the graft. So myself, the host, are the one I received this, right? Here, for example, in this case, let's go back. This recipient, or me, the host, I'm going to be attacking this graft that has been donated. I will attack it. Okay, this is the host versus graft. And this is what we know as transplant rejection, right? We always hear about it. I transplanted something, and then after some time, we'll see that it was not successful because the body starts rejecting it, right? Why? Why is it rejected? We mentioned, yes, we are different. The allograft and xenograft were different and stuff like that. The main reason is the MHCs. There's differences within the MHCs. Everyone has their own MHC, okay? The MHCs, they're kind of like these markers on our cells. If they're different, I'm going to be recognizing them as foreign, right? They're not self, so I'm going to be attacking them. Okay, here, this is what they're saying. They're saying immunorecognition of the antigenic differences. So there's antigenic differences. There are differences between the antigens, between me, the host, and the organ that has been donated, the donor organ. What is the donor organ called? The graft, right? And this will lead to immune response and all this, okay? What are these genetic differences that we're talking about? What are the genetic differences? This is the MHC. As we mentioned, the MHC is the genetic difference, okay? So when I want to transplant, I need to make sure of two things. I need to make sure that the blood is the same, blood grouping is the same, and that the MHC or the HLA, they call it HLA as well, which is going to be what? Human leukocyte antigen. So here, human MHC is also called the human leukocyte antigen. They're basically the same thing. MHC, HLA is the same thing. HLA is just saying that this is MHC for humans, okay? And there are many different classes. We're not going to be talking in detail here, but there's many different classes of this MHC. And it's going to be a combination of all. So it's going to be, I'm taking one class from this, another class from this, and it's going to be all a combination. That's what is going to be causing this variability. So it's highly polymorphic. It's going to be very different. And there's codominance in the ID. So basically, I get one from the mother, I get one from the father, and both of them are expressed. Okay? So it's based on genes, right? I'm getting one from the mother, it's based on genes. That's why in genetically identical twins, monozygotic twins, it's fine because they have the same genes. So they have the same, what, MHCs, right? HLAs. Okay. So here, this is what's showing. Here, I have the same as them, the same thing. Like they're literally the same thing. So it's accepted. Here is different, right? So it's rejected. Here, I have some sort of similarity. Like I have the orange here. So it's fine, it's normal, it will be accepted. Do you understand? So the more similarity I have between the MHC and the HLA, the more it will be accepted. That's the idea that they're saying here. Okay? Now this was the host versus the graft. My cell my body, sorry, will be attacking the donated organ. Why? Because of the differences in the antigens. What were the differences we mentioned? The MHCs or the HLAs, because of the genetics. We get one from the father, one from the mother. There are many different subtypes and subclasses. They're mixed together. They're highly polymorphic. They're very variable. Okay. Now, in the case of the graft versus host, what did we mention? The first one is the one that is attacking. So this is the graft. What was the graft? The donated organ, صح? So now the donated organ is the one that is attacking who? The host. And now it's the opposite. Now in this case that we went, if we go up, here, what I will see is that this will be attacking me. Do you understand here? And before, it was like this. The host is attacking me. Here, in this case, no, the graft is attacking the host. This is what we're talking about right now, the graft versus host. Okay? Why is this happening? Sometimes this is mostly happening in the bone marrow transplant. When I'm transplanting a bone marrow cells, right? In the bone marrow cells, we know that in the bone marrow, there's stem cells. There's also going to be the maturation of the B cells. We know B cells are going to be maturing here. Sometimes even the T cells are being formed here. T cells and B cells, both of them are formed in the bone marrow. B cells will remain and they're going to be maturing. So when I'm going to be donating the bone marrow or transplanting some bone marrow, I'm not 100% sure that I'm only going to be transplanting the stem cells in the whole hematopoietic stem cells, right? Sometimes I can by mistake get B cells or T cells with it. It's very hard for me to ensure, yes, I'm only taking stem cells. No, because you can't really do that. It's not possible. It's very difficult. So sometimes by mistake, you're going to be collecting these B cells and T cells. And because they are what? The B cells and T cells, they attack the immune system itself and whatever, and they are part of the immune cells, and they're attacking any foreign stuff. So now when this is going to be in my body, these T cells and B cells are not mine. They are from the donor itself. And when I took the bone marrow, I took some of the T cells and B cells of the person himself, the donor. So now when I put this in my body, the host, these are acting against me because they're not my own. Okay? So this is what they're saying. They result with when donor lymphocytes, called B cells and T cells, attack the graft recipient. Attack the recipient. Okay? So this is bone marrow cells or hematopoietic stem cells. They are taken from the blood and they're injected in the patient, right? In me, the recipient. So I take from the donor injection of the bone marrow, and then I give it to someone else. But when you're collecting the sample or whatever, are you 100% sure they're only stem cells? No, you can't be 100% sure. So that's why in this case, any type of transplantation, you need okay. And also this one is the T cells that we're talking about, or the B cells, if they're mature, obviously. So if they're mature, this is what they're saying. If mature T cells are transplanted with the stem cells that I'm trying to transplant, I'm trying to transplant stem cells. I'm trying to transplant the hematopoietic stem cells, the bone marrow stem cells, whatever. But by mistake, I get some of these mature T cells. By mistake, they're also coming with it. And then because of that, they will start to attack me. And this is known as the graft versus host. This is explaining it in pictures better. I'm taking hematopoietic cell transplant. I'm trying to get stem cells and transplanting it to the patient, bone marrow or hematopoietic stem cells. But sometimes by mistake, they will contain mature and memory T cells by mistake. When I get it inside in my body, they will start to be in my body, T cells that are not my own. They are the T cells of the donor. They will start to see, okay, this is all foreign. I'll start to attack it. So I'll start attacking. Okay? Now, the mechanism of transplantation, first, there's the sensitization and rejection. Okay, the sensitization is basically the recognition. If we remember in the hypersensitivity lecture, the video before it, the hypersensitivity we mentioned in the type 1 hypersensitivity, remember the sensitization and challenge. And the sensitization we mentioned is the preparation, is just preparing the complex. And here, it's also only preparation. What happens is that the antigens, right, from the graft, they're going to be transported. So the DCs, the dendritic cells, are going to be transporting any antigens from this graft that has been transported, and they're going to be going to the lymph node. They go to the lymph node. When they go to the lymph node, this is the sensitization only. In the rejection, this is where we know in the lymph node there's going to be T cells. The T cells are then going to be seeing the dendritic cells. We know T cells require recognition and they need to have antigen presented to them. They can't recognize antigen by themselves. So once they're waiting in the lymph node, once the DC comes, right, they're going to be then recognizing them, and they're going to be like, okay, this is not part of me. This is antigen. So they'll start to attack it. Okay? This is what they're saying. DCs will present it, and then they will stimulate the helper T cells. Okay? That's what's happening right here. And then after that, when the T cells are going to be stimulated, then they're going to be going back to the transplanted cell that we have, and they're going to be attacking it. Okay? In this case, this is the type of the host versus graft. So the first one we discussed, where the host is attacking the graft. Because here, this is the kidney, this is the donated kidney, this is the donation, this is the graft. The graft, the organ, the donation, it has antigens on it, it has its own antigens, right? There's going to be dendritic cells circulating everywhere. They see that, okay, they take the antigen, they go to the lymph node, they're showing it. The T cells, my own T cells, are going to be like, this is not mine. So they will go back to this donated organ, the graft, and they'll attack it. This is the host versus graft. When I talk about graft versus host, is different. When I talk about graft versus host, is different. Let's look at this. Normally, okay. What will I have normally? Normally, this is my self-MHC. I will have the T cell waiting, and then a dendritic cell or whatever will come, and they will present this foreign peptide, basically an antigen. They will present the antigen. As we can see, the whole antigen is fitting in. The T cell will recognize that this is not my own antigen, and will start to trigger and become what the T cell will be activated. But if it recognizes that this is a self peptide, if I said this is self, what happens? Nothing. It will be what? An energy or whatever, right? So here, they're saying my own MHC, so this is my own DCs. I'm talking about normally when I have sickness, just normally. Forget about graft and all of these things, transplantation, no. I'm talking about normally, I'm sitting normally, and I have some disease or I have some sort of antigen in me. My dendritic cells, okay, my dendritic cells will be collecting, as we know, they collect their antigen-presenting cells, so they'll be collecting any antigen they see. They got this foreign antigen. They're my own MHC. That's why we say self-MHC. They're going to be showing, okay, this is the peptide. And this is my foreign peptide. The T cell will be like, okay, this is not me. And then they're going to be what? Getting activated, okay? Now, in the other type, what we have is when we are talking about the transplant. Here now, this is the transplant, okay? This is the transplant case. Sometimes what I'll see that I have self-peptide. So in this case, this is going to be the peptide of the graft. And this is the graft, the kidney, let's say. We talked about the kidney, right? This is the graft. It has its own MHCs, right? It has its own dendritic cells, okay? These are supposed to be these, I think. Anyways, in this case, what we can see the difference is between these two. I want you to understand the difference first. The difference here is that the MHC is the one that is fitting. Here, what was fitting? The foreign peptide. Foreign peptide is fitting. Here, what I see is that no, the MHCs are the one that is fitting. And the peptide itself is not fitting at all. Look, not fitting. Not getting in the groove at all. Nothing. This groove is of the MHC. Nothing. I'm talking about fitting with the TCR here. So the only thing that's being fitting here is my MHC. In this case, what is fitting? It is fitting my MHC as well as my peptide. Now here, normally I will have the peptide and MHC normally. Yeah, because I have to recognize that this is my own self-MHC. We remember that in the positive selection, we're going to be supposed to have a proper recognition of my MHC, the groove of the MHC, and the antigen itself. So both things are required. So normally I will have binding of the MHC and the antigen. Here, what I see is that only the MHC is being here. Only the MHC are binding. هنا شو نشوف we're seeing that there's going to be both the peptide, this can you see the peptide, and the MHC. زين? Okay, so those are the differences. I will be discussing them now to see the differences more here. Okay, now so in these two cases, this is where the transplant is. Now I can have direct or I can have indirect allo recognition. So allo, we know the transplantation from the allograft, you remember? So that's the idea. We have direct or indirect. Okay, direct or indirect. Direct one is what? The direct is when we mentioned in the kidneys, they have their own D cells. Do you remember? These are the dendritic cells. The kidney, they have their own dendritic cells. The dendritic cells of the kidney, this is my kidneys DCs. The kidneys dendritic cells, they have their own LHCs and their own self antigens or peptides, يعني. Because I got it from the donor. It's from the donor itself. So they're going to be containing their own MHC, their own self antigen. Okay. Here, this is what we see here. Okay, that the MHC is being shown. So just the recognition of the MHC itself, just when the TCR recognizes that this MHC is not mine, it was enough for it to understand, okay, I don't, this is bad, I am going to be destroying it. Okay? Here, so that's right away the donor is going to be, that's what we're saying here. Look, when naive T cells recognize donor MHC molecules. So in this case, because they're their own, because the DCs I got are from the kidneys, they have their own MHCs and they have their own self-antigens. When they're being shown to the CD8, what's being bound is the MHC. So the T cells will recognize the MHC and they're like, okay, this MHC is not mine. Because you mentioned normally it will recognize what? Normally, as we can see here, normally I recognized the MHC as well as the peptide. Here, it's recognizing the MHC and the peptide, but here, once it recognized the MHC was enough for it to tell me, okay, you are foreign. So now this is what we see, that once this happens, it's going to be activating the T cells, which in this case is going to be CD8. CD8 cells are going to be activated. We know what cytotoxic T cells are going to be doing. They're going to be killing them right away. Okay? That's direct, because I'm directly from the own DCs. Directly from the patient's DCs is direct. خلاص, I saw MHC, it's not mine, that's it. The indirect allo recognition is going to be presented by my own self MHC, but the antigen is donor. The antigen is from the donor. I'll explain to you here. The direct, I mentioned that the donor's DC was there. The indirect is my own dendritic cells. They will go to this graft. They will see, okay, this is not part, they will just, the DCs will just collect any antigen. They will collect the antigen. They will start to break it down. So here's what they're saying, uptake and processing. So they will start to take any antigen, they process it normally. This is how it happens. They take an antigen, they process it, and they start to show it and present it on their MHC to present it to the T cell. The T cell will bind to what we mentioned. The T cell always bind to MHC and the antigen. It will recognize that the MHC is mine because it's what? Because from my own DCs, right? But then when it looks at the peptide, it recognizes that this antigen is not mine. It's not self-antigen. It's allogenic antigen. So now it will also be activated because it's not mine. It's not the antigen is not mine, and will be activated. But in this case, the CD4 cells are being activated. Do you understand the difference? The difference is that normally, this is the picture. Okay, let's just erase it. Normally, the T cells are going to be what? The T cells are going to be recognizing two things. This is normally. The T cells are recognizing two things. One is the MHC. Can we see the MHC? Can you see the green thing here? This is my MHC. Can we see it? The MHC. And secondly is my peptide. Can you see this foreign peptide? It's okay, the foreign peptide. So normally it will detect two things. In normal cases, it's myself MHC, and I will just see the peptide, and it's like, okay, this is a foreign one. Okay. In the transplant, it will be doing it in two different ways. Either it's fitting the MHC, and the MHC is not mine, and it's saying, okay, this MHC is not mine. I'll destroy it. I'll call the CD8. I'll be activated, and the CD8 will be activated and will kill it. Or it's what I have myself MHC from the dendritic cells. The dendritic cells, my own dendritic cells. So I have self MHC. So the difference here is directly in the direct allorecognition, the MHC is not my own. Why? Because it is going to be from the kidney's own DCs. Yani the kidney in this case is the donor. I got the donor, the graft, I mean. I got the graft. In the graft, I have the kidney itself, but also inside of it, I'll have the dendritic cells and whatever. Okay? The dendritic cells are going to be the donor. So this means that the MHC is going to be the donor. So to me, when it's going to be coming in contact with the T cell, the own dendritic cells, right? I'll see that the MHC is different than mine. So why would I react to it at all, right? بالindirect, no. It's my own DCs. They will come and they will start to take the antigen. They will break it down. They will present it. When the T cell will come in contact to get activated, it will bind to two things, the MHC and the peptide. The MHC, it will recognize it as myself MHC, but then when it looks at the peptide, it's foreign, so now it will be activated. But in this case, it's CD4. So direct allorecognition is CD8. The indirect is the CD4. Okay? I think there's a picture as well here. We can see that inside this kidney, this graft, I can see that there's donor dendritic cells and recipient dendritic cells. This is what I'm talking about. And the donor dendritic cells, the donor, this is the DCs of the donor. So the MHC will be not mine. The MHC will be the donor, صح? So MHC is different. And obviously the antigen is different. When my T cells will come to get recognized here, when they go to the lymph node, remember here, when it goes to the lymph node, okay? As we can see here, can you see this is the green one, the donor? It goes to the lymph node, it activates what type? CD8. Okay? Why CD8? Because it is going to be the direct way. The recipient is going to be what? The recipient dendritic cells, they're going to be taking antigens from outside, they're going to be getting them in, they will be processing them and breaking them down, and they'll recognize it. When they go, the T cell will be seeing that the MHC is mine, but the antigen is not. So now it will be activated to what? To the CD4. Do you understand the difference? This is the difference. So I think they maybe, hopefully you'll understand more here. Now, this is just explaining everything I have said, okay? Again, now once the CD4 will be activated, what they will do? It will start to release inflammatory cytokine. It will start to activate the B cells, right? It will do many of these things. Here, the mixed lymphocyte reaction here, we mentioned to avoid the transplant rejection, whatever. They will do many things. They can make sure that the HLA is the same and how that. But in the case of the graft versus host, where the graft is attacking me, I can do another thing where they're going to be basically taking T cells from my donor and T cells of my own. Okay, T cells from the donor are my self antigens, and they're going to be mixing them together, and then they will measure the toxicity of it. So if the T cells will kill these self antigens very much, I'll be like, okay, I don't want to do it. If it's a very small reaction, then yeah. So this is just a way we can measure. Now, immune mechanism of graft rejection. These are just going to be different steps. So there's going to be different features based on them. There's classification to the graft rejection. Could be hyperacute, acute, or chronic, okay? The hyperacute, from the name, is going to be occurring within minutes. It's going to be occurring really fast. And the reason is because here we're using circulatory antibodies. So antibodies are already circulating. When I got this graft, what happens is that the rejection is from the antibodies itself that are going to be circulating. Again, the same way, we remember the circulating antibodies are through what? They're going to be through the, they're going to be soluble, right? So they're soluble and whatnot. So they'll bind to that and then they're going to be causing what the vascular endothelium tissue. Remember that there's going to be the inflammation, and then the antibodies, once they bind, they're going to be activating the complement and natural killer cells, right? And the acute rejection is days or weeks. So hyperacute was what, hours, minutes? The acute is days or weeks. Why? Because here in this case, we're using T cells and antibodies. The first one, hyperacute, was within a few minutes because I already have circulating antibodies. For acute, where it takes much more, a bit more time because we're making use of T cells as well. Again, the same way, CD8 cells will start to destroy the graft directly, or in CD4, they're going to be secreting cytokines, the same way that we have mentioned. And then again, the antibodies, because we mentioned there's use of T cells and antibodies. The T cells will do their action through this way. Antibodies, how they're going to be working? Through complement activation. They will activate the complement by the classical pathway, and then they're going to be doing the same way of the recruitment and whatnot, right? And the chronic rejection, no, it's going to be months to years. So we can see as we're going, hyperacute is minutes, occurs within a few minutes. The acute is going to be occurring within what? Minutes, hours, days to weeks. Chronic rejection is going to be longer, months to years. And why? Is because it's only using T cells. And we can see that the time is correlated with what is the type of immune response based on what, right? Here, we can say that the chronic rejection is T cells only. That's why it takes a lot of time. And in this case, the T cells will start to, mainly we're talking about secreting cytokines, because if it was CD8, mostly it will be, I'm destroying it right away. So it might take fewer time. But here we're talking about mainly is the secreting of the cytokines. So we're using CD4 here, okay, secretion of the cytokines. And then this will stimulate the proliferation and all of these things. But also it will, another thing that's unique is the fibrosis of the graft. It will cause proliferation of fibroblasts. So it's going to be forming a lot of these fiber, and you'll see that the graft is going to be very fibrous in nature. And then also narrowing of blood vessels. So these are the unique, the two different unique things about them. The acute, hyperacute is going to be what? Within minutes. What is being used? Circulatory antibodies. Okay, and then the same way circulatory antibodies bind to the antigens, activate the complement, the same way we took it in the hypersensitivity. If you haven't watched that video, watch it, okay? The acute rejection, we mentioned, is going to be within days to weeks, and the reason is because we're using both antibodies and the T cells, okay? And we mentioned the T cells, what they're going to be doing, the CD8 and the CD4, and then the antibodies, again, they're going to be activated by the classical pathway and whatnot, they're going to be doing the same thing. Chronic, no, it's going to be only the T cells, specifically the CD4, and they're going to be secreting cytokines, and it's over months or years because only the T cells are being used, no antibodies, and the T cells, specifically the CD4 cells. And then we mentioned there's going to be fibrosis of the graft and narrowing of the blood vessels. Now here, how can I prevent and treat the graft rejection? Many of the ways we mentioned, okay, first, the HLA typing, we need to make sure the same MHCs, we need to make sure the blood types are the same, whatever. But if I want to prevent all of this, the rejection, I can use immunosuppression. Immunosuppression, these are drugs that are going to be, from the name, suppressing the immune system. They will suppress the immune system, and they will either decrease the amount of the T cells, or they will inhibit the T cells from being activated. So now the T cells will no longer be able to recognize that this is a foreign antigen, or the many complications that we see, how the T cells are recognizing that this is a graft and whatnot. So now once they are going to be suppressed, they won't be having the ability, but obviously, to recognize that this is a self-antigen. But obviously this comes with a cost, because now you won't be able to recognize any of the antigens from the world. So you will be more susceptible to diseases and whatnot. So that's also a problem, okay? But usually we use immunosuppressants to avoid all these rejection processes. All right, so this is the end of the video. Thank you very much. If you have any questions, please write them down in the comments. And thank you very much. I hope you have understood.